talk is by Erick Rodriguez, Interactive Identification Key to Identify Third Instar Larvae of Anastrepha. Please.
Thank you, Severin. Good morning, everybody. So I will be talking more about what we have been doing in terms of the larva morphology in order to create an online interactive key. So the genus Anastrepha occur in the American neotropics and subtropics. It comprises more than 328 species. So among these species, we have 7 pest species. One of them established in Florida since 1965, and the other one, Anastrepha ludens, which have multiple incursions in Texas and a few in California. And they attack mainly commercial fruit, such as mango, citrus, papaya, guava, etc. So over the last 150 years, the taxonomy of this genus is based on the female adult. On the other hand, immature are poorly known. So despite of the economic importance of these pest species and the high diversity of this genus, we have some limitations. So in the last 90 years, 7 keys have been created, and mainly they have been dealing with pest species. One of the best dichotomous key was that of State 1990, which include 13 species. But I have some problem there because 3 species in the Fraterculus group, for example, Anastrepha suspensa, Anastrepha fraterculus complex, and Anastrepha obliqua, they overlap and it's difficult to distinguish. And the other problem with this kit was that 2 species in the strata group are difficult to distinguish. These 2 are Anastrepha strata and Anastrepha bistrigata. So the online interactive key that is still available is that of Carroll et al. 2004. But again, it has also some limitations. So these 2 keys are out of date. So so for this project, the target species are those 16 species, including in Carroll et al. Those 11 species that Diptera et al. And Frias et al. Described in the last 10 years, and those 11 species recently described by Erick Rodriguez and et al. So the goal here is to compile all the data, all of these larva descriptions, and incorporate this into a Lucy Builder platform. Also, in Rodriguez et al. 2022 found new characters. So these characters and new characters today will be incorporated into this key. And finally, create a new interactive key specifically for the genus Anastrepha. So here, behind this, there is a lot of work, multiple trips to South America, Panama, Central America. I did a lot of the collecting of both larvae and adults. And the good thing here is also that everything we are looking at, every immature we are looking at, we are using DNA just to confirm the identity. So the other thing is to prepare a specimen and illustrate it using a scanning electron microscope and light microscopy. So, you can see here, there are many, many characters on the head. So in the head, but also we have to look at characters on the thoracic spiracles, also in the posterior spiracles, and also along the dorsal and the ventral part of the larvae. So one of the results we have is we have a data matrix built in using Lucy Builder version 4. You can see here we have all the characters and character states and those across the different taxa. So this key will be hosted at the U.S.A. APHIS Identification Technology Program, and it will have some good components. So the user will be able to see fact sheets, so that it will have, for example, diagnosis, how biology, distribution, economic importance, status of knowledge, specimen examined. And this is one of the examples that the user will be able to read and also to help get to narrow down the identification of any larvae that we are including in this key. So also the user will be able to search for any glossary or any structure that have some doubt. So it will be able to see. So here we are using those glossary published by White et al. 1999, some that we adopted from Courtney et al. 2000, from those that we discovered in 2000 by Rodriguez et al. 2021, Steck 2022, and recently those characters recently discovered by Rodriguez et al. 2022. So another good feature here in this website is the gallery. So the user will be able to search images by body part, by species. So that would be helpful to the user go across taxa, for example, and compare body parts and help you to go faster on this identification. So the key Excel have 41 taxa and 23 characters. That means 46 character states. So as you can see here right now is we are working on the Anastrepha key, but in the future we are planning to develop another key for pest genera key, pest species key, Dacus, and NOM. So we'll go through different genus. So this is where the key will be available for the user, will be soon available. So will be published pretty soon. So okay, the conclusions. So of those 23 characters, 14 are found on the pseudocephalon, on the head, and they are very, very useful to identify species to species. Among those 14 characters on the head, things are the best character to distinguish species. So among these 9 characters that are the best, 9 are for the first time used in this key. And you can see here, this is what we call the preolar organ. Are some cuticle processes around the preolar organ. And this is another character that we recently discovered, which we call it a cuticular cone-like cuticular processes. So here, we can see some of the other characters we are using. For example, so we have preolar organ and preolar lobes that are fused or can be separated. So another interesting feature is on the ventral surface of the map hook. So we have discovered that some have a medial carina, some species have very rough surface, the ventral surface of the map hook. Also, we use the position of the preolar organ. So some larvae have the preolar organ lateral to the map hook, or can be anterolateral to the map hook, or can be anterior to the map hook. So, again, with this study, we found similar situation, like in that key of Steck et al. 1990. So some species, like Suspensa, Turpinia, and Senilde, are very difficult to distinguish. And here the problem is because some of these species, the larvae of these species, have partial description, or in the publication we have only we don't have good SEM images to look at the characters and capture the data. So future edition of this identification tool will be include additional species of Anastrepha. Of course, the characters they can be changed, and also the coding of these characters can be changed. So with this, thank you very much. So I thank too many collaborators and student, UF student, that they have been helping us with the images, and many collaborators in the US, you can see here, and many collaborators in South America. With that, thank you very much.
Does anyone have questions?
Yeah, SEMs gold coded show really rounded characters, but the users will have their flies in methanol and maybe dirty from plants. Or so, will you include our section with maybe some film how best to prepare the larvae, use light angles, just techniques to see these characters?
That's a good question. Look, it's something that it could be included in the website. You know, for example, how we can prepare larvae for optical microscope or SEM, and also how to look at the larvae under dissecting scope or compound scope. So we recently in Gary and I we have been using the compound microscope using 10x to look at those feature oral ridges or accessory plates. You take the intact larvae and let air dry it for a few minutes or two, and then you place it under dissecting scope. You will be able to look at those features.
We have another question actually, because
So your question is, when could be available? That's a good question. Gary's here, and Gary and I, we are in that same project. So personally, I'm trying to get a good pest general key by the next fruit fly meeting that will be in Jamaica. That's my goal. Gary and I will be working on it. If we have something before that, we'll see. But this is a good question, Bill. So thank you for the question.
well, thank you, Eric.